So fun, even though I can't visit the campus. It's such a pleasure to actually tell you a little bit about the work in my lab. MK is right, I really enjoy questions, and the virtual format kind of compromises that in some way. It's hard to sort of connect with the audience, but what you'll find is I actually have too much material for a regular sort of 45-minute seminar. So I always tell people I'll pause at several times, kind of natural breakpoints, and maybe we can field some questions. MK can read the questions in the chat box if there are some at a couple of midpoints, and never feel like that's going to make me go over. One of the pet peeves of mine is speakers that run past time. So I presume this is about 45 or 50 minutes. So I'll make sure I stop at 10 after, maybe a quarter after the hour, and make sure there's time for some questions at the end as well. And if I don't get through all the material, I'll just zip right to the conclusions at the end, and that's fine. Just a couple of quick disclosures. I'm happy to talk about startup companies. I've had the good fortune of being involved in two of them, Irradiate and Immunovalent. The work I'm going to tell you about today is most relevant for Irradiate. That's the one that's actually doing pretty well. It's based in Irvine, based on some technology out of my lab from probably 10 years ago now. It takes that long to sort of develop them. Immunovalent, I started at WashU, and to be honest, that one's on kind of life support right now, but it's still viable. I like to start with acknowledging everyone at the beginning and give you an idea of the work and who's done it beforehand, actually. Lots of collaborators. My lab is just so dependent on working with others. I think that'll become pretty obvious as we sort of move through some of the work. Everyone sort of now has Zoom pictures of their lab. This is one of our earliest ones, probably from back in April. Usually have about 10 people in my lab. It's kind of a blend now of professional scientists, postdocs, and graduate students. And I kind of like that blend now, as long as I can sort of maintain it. It's been nice, and including a new position. I have a scientific writer that we're trying out for a year. Nuala was a postdoc in the group, and she's really interested and talented in writing. So I'll tell you a little bit about some of the work from collaborators, a couple at WashU, Kathy Weilbacher and Dan Link, that's in the bone marrow space. Ryan Fields was at WashU, and Sep Galami at UC Davis. They're both surgical oncologists, and that'll become obvious why we collaborate with them as well. And the work, for the most part, that I'll show, two people that are still in the lab. Drew is a postdoc. She's just finishing up. She'll finish in a couple of months. And Venkatesh started as a postdoc. He's now a project scientist. And then some of the work has been done by others in the lab. So I think you would definitely call my lab an organ on a chip lab. I still am trying to decide if I'm a fan of that term, but I think everyone kind of knows what it means. It's basically tissue engineering. My formal engineering training is in chemical engineering, so I tend to be really interested in transport phenomena and fluid mechanics and transport of mass and things like that. So we call our lab the Transport and Tissue Engineering Lab. We work in a variety of areas, just to stick with the sort of on a chip idea. We have vessels and bone marrow, and we work in the heart. That's actually the largest piece of funding I have. Tumor biology and some migration of cancer cells and immune cells as well. There's no way I'll have a chance to tell you about all those projects. And to be honest, I won't even get to some of them. What I'll spend most of the time telling you today is the vessel on a chip, just because it's really a core technology. And that sort of led into the bone marrow on a chip. And if we have time, I'll tell you a little bit about what's going on with the tumor and maybe some basic science applications that we worked on when my lab was at WashU in St. Louis. So I kind of like to start with this figure. Some of you in the audience may actually recognize this image. It's become really quite popular. It's a review, it's a paper, it's an image from a review paper that's now, gosh, 11 years old. But it sort of captures the tumor microenvironment. And so it was really one of the first images that was so, I don't know what it is about the image that everyone likes. I guess there's just so many different cell types, and it's kind of messy looking. So if you're interested in tumor biology, it kind of captures lots of things. The tumor cells are there, of course, and in abundance. But what's so important is all these other different cell types. And I don't expect you to sort of read or look at them, but there's blood vessels and stromal cells like fibroblasts and plenty of leukocytes. I really like this picture because it also includes the bone. I didn't actually piece this in. That's actually part of the figure. And it describes how the bone marrow actually communicates back and forth. So the tumor can actually draw immune cells from the bone, but tumor cells, especially in breast cancer and prostate cancer, tumor cells can actually go to the bone and reside there as well. So there's a really interesting communication. So if you're sort of in my position, or you're a trainee in an organ on a chip lab or a tissue engineering lab, and you're interested in engineering tissues, this is just such a fundamental question that I think you have to ask yourself all the time. What features, in this case, of the tumor microenvironment are absolutely essential for you, for your particular problem? You won't be able to model everything. This goes for other organs as well. The tumor, you just can't do everything in an in vitro setting. So you have to pick and choose a bit, and you'll see how we've made some choices as well. So I've highlighted kind of 4 categories of kind of decisions you'll think about. Thinking about tumor cells, the source of them. Are they going to be primary, which means tumor cells right from the patient, or tumor cells that maybe are cell lines that represent human biology, or they could be tumor cells from animals, but you have to have a source of tumor cells. The vascular network and the vascular supply is something that my lab has spent a lot of time thinking about. It's sort of inherently interesting to me. I mentioned my training as a chemical engineer, so this is how things are transported in and out of the tumor microenvironment. The extracellular matrix, it's not really shown that well in this particular image. You see it sort of out here in this sort of brown color, but it's basically the supporting proteins like collagen and laminin and other things. We pay attention to it, but I don't have it bold-faced here because we don't really specialize in that part of it. We do have to have a matrix, but you'll see I don't really spend a lot of time manipulating it. And then there's all these host cells. There's a whole bunch of them. I list stromal, which is another name for like a fibroblast. It's sort of the maintenance worker cell of the tumor microenvironment. Lots of different immune cells. I just have captured them just with the word immune, but there's 10, 12, 15 different kinds of immune cells. And then cells that you can actually recruit from the bone marrow itself. And I mentioned that's why I like this figure. So I think these themes will all come through when I start thinking about how we sort of model the tumor microenvironment and how it interacts with the bone. So this is sort of a concept that I've been thinking about for probably 5 to 7 years now. I don't think it's a particularly novel concept. Maybe it was back then, but now lots of people are thinking about it. And I don't think it was my lab that really drove this. I think it's just a natural extension of the organ or tumor on a chip field. It was the idea that could we use this for personalized tumor or cancer therapy? So the concept is pretty straightforward. I show here breast cancer and colon cancer just because those are the 2 that we've generally worked with, but this could be applicable for any tumor. And the idea is, as a clinician, you're faced with maybe a choice on what type of therapy to use. Sometimes there's not a choice, there's just one, but many times there's different choices. So how could we be, I guess, smarter about making that decision or make it more personalized? So the idea is you would take a biopsy. You take that biopsy and you put it into some type of culture system. This is a picture of a device from my lab or a schematic of it, but it could be really any type of ex vivo or in vitro type of culture system. The tumor spends some time in the device. I show it here with a vascular supply, and you'll see why a little bit later. And then at some point in time, you expose that tumor to some type of therapy. It could be a chemotherapy, could be an immunotherapy. And you watch how it responds, and maybe it helps you make a decision to sort of optimize that sort of personal therapy. So this is just sort of a general schematic of what kind of drives many discussions between myself and people in my lab. So if you're going to do something like this, step 1 is the perfusible vascular network. I showed just a schematic of it there. So I like to start with what we've done in the lab now with the sort of what I consider the core technology. So back in 2000, I don't know, 2001, 2002, almost 20 years ago, I was sort of transitioning into this field, and a collaborator of mine that I still work with at UC Irvine showed me how to make in vitro blood vessel networks, and they're relatively straightforward. This has become really and continues to be a core area of expertise in my lab. We actually teach undergraduates when they come into the lab how to do this. It's pretty reproducible, and it highlights a number of important themes that I'll mention. You need a source of endothelial cells. These are the cells that actually will make the blood vessel, at least make the inner lining of the blood vessel. And then you need a fibroblast or a stromal cell. And so we generally use endothelial cells from cord blood. You have lots of different choices. I'll also show you endothelial cells derived from stem cells. And we started out using lung fibroblasts, but we've now made vascular networks out of probably 10 to 12 different stromal populations. So you don't have to have any one particular kind. Expand the cells in the lab, put them together, and then randomly distribute them in some type of hydrogel. We tend to use fibrin. You can add growth factors. You don't really need to. The stromal population tends to produce the growth factors that are necessary. And I like to sort of stop here and mention that it doesn't really seem like there's a lot of tissue engineering going on here. This is sort of a theme that MK probably remembers from the lab too, which is to sort of try to avoid the temptation, I think, as a tissue engineer to over-engineer your system or over-dictate it. The cells, like endothelial cells, are programmed to make vessels. They're actually quite good at it. And what we're doing here is just giving them kind of the starting materials and an environment that then allows them to do it. So we kind of refer to it as a biology-directed approach. We let the biology do most of the work, and we provide sort of just an environment to get them started. So they'll form a vascular network as early as 4 days. I should probably update that slide. And they can be stable for up to a month in culture, depending on how you treat them. It's easy to label them. You can put in endogenously expressed fluorophores, or you can label specific proteins like CD31 as a membrane protein after the vascular network is formed. And it's quite easy then to visualize them. This image is now probably a decade old, but we see this image all the time in the lab. It's pretty obvious what I'm looking at. It's basically a vascular network. It's from human cord blood derived endothelial cells. I think these are lung fibroblasts. And this is probably a 7-day-old network. You can see the scale bar down here, 100 microns. So most of the vessels in this state are probably 10 to 15 microns. They probably really do resemble a capillary. Sometimes they get thicker, and I'll show you certain conditions where they actually get a little bit larger. So these were more or less the vascular networks that my lab was growing for maybe 5 to 7 years, from maybe 2003 to 2009. We were using them really to create implantable tissues, to kind of pre-vascularize tissues for implant. And at some point in time, I like to tell the story this way. I was talking to my collaborator, Chris Hughes, and I said, gosh, these vessels are kind of interesting, but in vivo, there's actually flow in them. So how sort of biologically relevant could they be? And that's really driven what my lab's been working on for probably the next decade. So this image I'm showing you here, there's actually no flow of cell culture media or blood or anything like that in it. So it's just really the blood vessels themselves. So I left this in my seminar, and I think I'll probably continue to do it. There was a grant opportunity in 2009. It was called the Challenge Grant. So if there's any faculty in the audience that were running labs then, you probably remember these challenge grants. It happened when the transition, when Barack Obama was elected and the economy was tanking with the financial crisis, and there was a huge stimulus package. It was about $800 billion, which actually by today's stimulus package seems small, but it was huge at that time. And many people don't remember, but the NIH actually got $10.4 billion, a huge chunk of money. And they were supposed to spend that in about 2 years on the sort of scientific infrastructure to really kind of try to stimulate the economy. I think it was a long-term investment. So there was all of these opportunities. One of them was called these challenge grants. And this is the one that we applied to 11 years ago. The ES here stands for Environmental Sciences, which I had nothing to do with. But what I did notice in this particular challenge call was they were interested in miniature multicellular organs for high throughput screening. in this case for chemical toxicity testing. But again, that wasn't particularly interesting to me. It was the multicellular organ idea. And we were thinking at that time about, could we actually get flow in these vascular networks? That would be such a basic concept for any type of multicellular 3D in vitro organ. Every organ outside of maybe your cornea in your eye has a blood supply. And so it seemed like an interesting sort of proposal. So it actually got funded. And I mention it because it really has dictated my entire lab. My lab shifted away from doing really primarily pulmonary work. So it's an interesting example of how your lab can potentially pivot and move into new areas over a long career. And you have to sort of look for these opportunities. So this is actually some images from that actual grant application. We actually showed a picture of a capillary network. It'd have an arteriole and a venule, blood vessel, little capillaries connecting them. And we thought we could do this in a microfluidic device. The arteriole would be a microfluidic channel, and the venule would be a microfluidic channel. And in the middle, we would actually put our endothelial cells and fibroblasts, get them to make a vascular network, and then try to convince them to actually connect or anastomose with these microfluidic lines. And if we could do that, the high pressure fluid here would flow through the vascular network, and we would have a perfusible vascular network. So we don't really use a design like this, but we talked about it being biology directed, high throughput in nature, so you could potentially either screen drugs or chemical toxicity. We were going to use an optically clear platform, so it would be real-time monitoring would be available. And we thought it was pretty cool because this could be really quite a dynamic model. We could look at blood vessel growth or regression or all kinds of possibilities. And as you'll see later, we could add functionality to it. We could turn it from a generic vascular network into something that was actually more functional. So a couple more slides, and then maybe I'll pause and see if you guys have any questions about this sort of vascular technology. So be thinking about maybe typing in some questions. This is one of our early devices. It just highlights a couple of features to it. This one I would say is maybe medium throughput. Each one of these little diamond-shaped devices, which we actually still use today, some minor changes, is sort of a separate tissue. We would actually inject through this port on the left a fibrinogen solution with endothelial cells and stromal cells. If you put a little bit of thrombin in, the fibrinogen clots and forms fibrin, but it's liquid for long enough that you can actually inject it through from left to right. And in this case, there's 12 tissues. Then the fibrinogen clots to form fibrin, shown here. And the endothelial cells and fibroblasts do their thing. They form their vascular network. And then a postdoc in the lab, I don't know if you can see her picture, Monica Moya, was the one to sort of figure out how to sort of get them to anastomose. The vessels actually like to grow against flow. And so we had a little connecting port, for example, right here. This would be the high pressure side. So fluid, in this case, cell culture media, would enter this small chamber, percolate across the chamber to provide nutrients, and exit down here. And when the vessels were developing, they would like to grow upstream toward the high pressure side. So it took her a while to figure out how we were going to get anastomosis on both ends. And all she did, it's actually really simple in hindsight, she just switched the hydrostatic gradient. So this was the high pressure line for 24 hours. And then she would switch it and she'd make this the high pressure line down here for 24 hours and then switch it again. And then the vessels would actually grow up and connect with both ends. Once you got connection, you could actually leave one side, the high pressure side. So we still use hydrostatic heads to this day. You can use microfluidic pumps if you like more controlled flow, but microfluidic pumps have their own problems with air bubbles, and it's just a little more complicated. So we actually think if you have a big enough reservoir, you can maintain pretty decent flow through the networks using this way. So we published this paper about 7 years ago now. And this is one of the early videos. We have all kinds of them now, but this one is kind of interesting for lots of reasons. Monica used to tell me that she could actually tell whether or not there was flow going on in the vessel network just by what the vessels looked like. And so you'll see, based on this scale bar, 100 microns, that some of the most active vessels here that have these fluorescent microbeads flowing through them are thicker the wall is thicker and the diameter is larger than what you would consider a typical capillary. So I usually refer to this as a microvessel network. So the high pressure beads are flowing through a media up here. They enter the network, they flow through, and you can see the flow pattern is really quite complicated. It's generally going top to bottom and generally going left to right. So if you look at my laser pointer, you can see some of the vessels are actually exiting this chamber and entering a neighboring chamber on my right. Couple of problems you can see here. There was sort of a leak at the top, right? You can see a bunch of beads that are just collecting in the fibrin gel up here. So I don't have a tight anastomosis. And that was problematic for a number of years. We spent a lot of time and energy trying to patch that leak, which I think we've now, we accomplished that. It took a few years though. And without that, we really were not able to manipulate much pressure or flow. You can imagine if I increased the pressure or tried to change the flow rate, I could just basically blow out that particular leak. So that was kind of problematic for some time. So let me let this video run, and MK, maybe I'll just pause here and see if there's any questions on kind of the core technology of making this vascular network.
Sure, sounds good. Are there any questions? We've got about 60 people who have logged in for this presentation, which is awesome. If there are questions from any of our students or visiting faculty, please put it in the chat. I'm happy to moderate.
If there are no questions in the chat now, it's fine. I can just keep moving on, but this is kind of where I usually kind of break and see if there's questions.
MK, I put a question in the chat for you.
Thanks. So Joel Berry has asked, he says the hydrostatic pressure head looks really small. Is the gradient created using this method physiologic?
Yeah, it's a really good question, Joel. So the absolute pressure is not, the absolute pressure is quite low. It's like maybe a centimeter of water, right? Which is not physiologic, but the flow rate that it generates across this vascular network is physiologic. So I up here, you can see my laser pointer, I think. So physiologic velocity is somewhere between 0 and 4,000 microns per second, quite a wide range, but the mean is about 690. So average flow through a capillary network is about 1,000. So we can generate physiologic flows, but the absolute pressure is not.
There's another question. What types of growth factors are used in your system?
Yeah, I mentioned that you could add growth factors if you would like to. The classic ones are VEGF and FGF, fibroblast growth factor, but we don't actually add those anymore. You can supplement if you'd like, but the stromal cells that are in here that are added with the endothelial cells are sources of growth factors, and we tend to believe they will make the amount of growth factor that you would need to sort of create the stable network.
Awesome, thank you. And then I think our final question for this round, oh, no, sorry, people keep sending them. We have a question from Dr. Ben Wren, who is in the Department of Surgery and works with endothelial cells. He wants to know if you can make the vessels resistant to arterial pressures.
I don't know. It's a good question. We've never raised the pressure that high. I would doubt that these would sustain an arterial pressure. That said, we've certainly been interested in that problem, mainly from whether or not you can make these vessels vasoactive. And the startup company that's based on this technology, they have an SBIR grant that I actually wrote, and it was to actually put smooth muscle cells into this culture to create something that would more resemble an arterial. I don't think our goal was ever to get pressures that high, but to see whether or not drugs would change vasoactivity. So I think that's really where that's at.
All right, thank you. Just a couple more, if you don't mind. How frequently do you need to rebalance the fluid columns in order to achieve relatively constant flow rates?
Yeah, you can imagine it depends on the size of those fluid reservoirs. If it's a relatively simple experiment, we just use pipette tips, but those are actually pretty small volume. These larger cryovials, we can usually maintain. We actually have been thinking about this for a different device a lot the last couple of months, And it's pretty easy to maintain a constant shear, a relatively constant physiologic shear for about an hour or two before you have to top it off. If you don't care about maintaining a constant one, you could probably top off those reservoirs about every 24 hours.
Okay. And then have you hooked up a pulsatile flow system into any of your devices before?
No, we haven't. It's a very good question. Pulsatile flow would really only exist in larger vessels. Once you get down to capillaries or even small arterioles, the pressure fluctuations that you see from the pumping heart are actually damped out completely. So that's a feature we haven't, I guess, needed to be worried about if we were just thinking about the capillaries. Yeah, really good questions.
All right. I think in the interest of time, let's head on, and we can always circle back around if we need to.
Yep. Oh, that sounds great. I'm good. Okay, so a few more thoughts on endothelial cells. I mentioned we've made endothelial cells from iPS derived cells. Remember, this technology wasn't even around when I started making vessel networks, but now it's actually kind of commonplace. They're a little problematic. They're not great. I'll show a picture down here, which many times you'll see in seminars are always the best picture you ever got. So it's a beautiful vascular network that Yosuke created, graduate student at WashU, but difficulty sort of getting reproducible networks. So we just demonstrated we could do it in principle, but we've never actually replaced the cord blood derived endothelial cells. And there's lots of different protocols for doing this. I do like this. This is a video that runs that kind of shows that self-assembly process and the day numbers in the top left. So these are iPS, or stem cell derived endothelial cells that are constitutively expressing M-cherry in the VE cadherin, or junctional protein. So you can see over the course, I think it's 14 days, how they spontaneously form this vascular network. Not all the endothelial cells are actually incorporated into the network, but it does give you an idea of how that works. The anastomosis point, this is that pore. It's a microfluidic pore. This would be the microfluidic line up here, and down here is the chamber where the vascular network is forming. And I like this image. You can see the junctional proteins from that marker, the VE cadherin M-cherry. And you can see how the endothelial cells basically coat this channel, and they sort of rise up and form what looks kind of like a funnel. And depending on how long you keep them in culture and a number of other conditions, they will actually grow up and begin to line the actual fluidic chamber. That can be good or it can be problematic. It depends on the dimensions of that fluidic line up there. Other things that you can look at in these vascular networks are certainly staining for something really basic, like are they making their own basement membrane like laminin? So we have a number of images like this. And this confocal image also shows nicely the sort of luminous structure. Here you can see it down here as well. And this is a picture again of one of our sort of bigger successes using the iPS, or stem cell derived endothelial cells. I show this one here, not because the iPS networks work all the time. I mentioned they can be a bit problematic, but I like this because it sort of highlights a slightly different shape here of the pore. And that was really key for us patching the leak. So now all of the dextran coming in here actually enters the vascular network and then empties in the low pressure line down here. Okay, enough on blood vessels. So we still work with some vascular problems only, but what we spent a lot of time doing is now incorporating function. So I'll move on to what we have spent now probably for 5 years with a variety of different grant opportunities. Has allowed us to build a vascularized bone marrow model. And this was really brand new to us. We were really motivated by our interest in breast cancer dormancy in bone marrow. It's a huge problem in breast cancer, but it's also very interesting and very challenging. Just by the definition of a breast cancer cell going into bone marrow and becoming dormant, you can't find it. It doesn't proliferate, it basically hides there, and trying to understand what causes it to come out of dormancy at some point later in time is just continues to be a real challenge clinically. So when I started this, didn't really know much about bone marrow. You probably saw my background. I went to med school many years ago, but didn't do any clinical training afterwards. So I tell people I usually know enough about each organ system to be slightly dangerous. So I did I do know what bone marrow is, and I knew sort of the basics of it, but I rapidly started knocking on doors when I was in St. Louis at WashU. There's of course a very strong med center there. And not surprisingly, there was a bone marrow expert, and that's Dan Link, and I mentioned him in the acknowledgements at the start. So Dan Link really studies blood-borne cancers, but that provides a ton of expertise in bone marrow. So we talked a little bit about modeling bone marrow, and he was really influential. He said, you know, Steve, if you want to model bone marrow on a chip, he said, the one thing you have to be able to do is you have to demonstrate that you have a viable hematopoietic stem cell, that it's stable and that it can actually differentiate into all of the major lineages. If you can show that, then you've got a model of bone. And that's essentially what we tried to do. So if you start digging through the literature and learning about bone, you'll see that it has been described that there are different niches. One of them is called an endothelial niche, which is near bone. And one's called the perivascular niche, which tends to be right near blood vessels. And I'm not sure I'm a big fan of it. The main problem is the perivascular niche is not where all the blood vessels reside. The endothelial niche has blood vessels as well. And so although the name's confusing, the easier way to think about it is these two niches perhaps have just different stromal cell populations. And that's what we ended up modeling. So the endothelial niche has the osteoblast. Sorry, my laser pointer is jumping around, shown here in yellow. And the perivascular niche has sort of a mixed stromal population that includes mesenchymal stem cells as well as fibroblasts. So we went about creating this. All of the images that I'll show you and stuff are a paper that Drew worked almost 5 years on, and it's in final review at Science Advances, but we've deposited it in the bioarchive as well. So we were interested in modeling these two niches. There was some hints in the literature that cancer dormancy might prefer one of these niches, but they clearly communicate. They're in close communication in the bone marrow. So Drew came up with a design here where we could actually create the niches on a microfluidic device where they would communicate across these little pores where my laser pointer is. And then she had the clever idea of actually putting a third compartment down here, which we would keep empty. But as soon as our bone marrow was mature and ready to study, we could, for example, load it with cancer cells and examine where cancer cells might migrate. And I'll just show you some, if I get to it, just some very simple pictures. So the strategy is almost exactly the same here. We put in endothelial cells with a stromal cell population. Those stromal cell populations are different between the two niches. And now we introduce the CD34 positive hematopoietic stem cell that is so central to modeling bone marrow. We get these cells from cord blood. It's the same source as our endothelial cells. Over the course of maybe 4 to 7 days again, the vascular network forms. And I think Drew put in some very fancy animation here. We can digest it at some later point in time with an enzyme called nattokinase, remove the fibrin, and take the cells out. And that becomes an interesting way of then examining what on earth you have in bone marrow. Bone marrow is very messy. There's many different kinds of cells. So this is where we rely heavily on flow cytometry to find out what kind of cells we have. I just show here that many of the cells are CD45 positive, which is just a marker of leukocytes. But we also take the cells out and we can do the sort of poor man's version of a bone marrow transplant. We don't get enough cells to actually put these in a mouse to see if it would repopulate the bone marrow of a mouse. But you can actually look at the morphology of how the colonies that might form and determine whether or not you have a hematopoietic stem cell. So I'll just show you some pictures. I think they're worth more than just lots of graphs and data. You can look at the manuscript that's again at the bioarchive if you're interested in all of that. But here you'll just see the vascular networks that form on both of the niches, the endothelial and the perivascular. I think just by bright field, you'll probably say, wow, they look different, and they do. They do have a different stromal cell population, and they behave a little bit differently too. Here we've just labeled CDH5 as VE cadherin. It's just one of many labels to look at the vascular network, and that also sort of highlights there is a vascular network in both of the niches. That's pretty reproducible, but they look different. The one in the endothelial niche with the osteoblast usually looks a little murkier, a little muddier. it's a little more permeable to dextrans that I'll show you as well. And that could be considered a physiologic feature of that network as well. These are perfusible. This is actually a video running, so you'll have to look carefully as the red 70 kilodalton dextran slowly fills up the vascular network from the top and then empties out into the bottom low pressure line at the bottom. I'll let that run a few times so you can actually see how that works. And you can also see there's sort of blushing of the red in the matrix part, and that would be indicative of this vascular network over here on the endothelial side having a higher permeability to 70 kilodalton dextran. So for those of you that aren't as familiar with blood vessel, a sort of normal standard type of vascular network in a human would be pretty impermeable to 70 kilodalton dextran. So that's a very common molecular weight that we use. The bone marrow vessels are more permeable. There's a lot more cellular trafficking, and we would expect the permeability to be a bit higher. So it's quite easy to actually measure this permeability. I won't go into the math to do that, but it's pretty easy. And what we find in the endothelial niche is a permeability that's probably 4 to 5 times that that's in the perivascular niche. And the perivascular niche looks more like the permeability when we use a lung fibroblast to create the vascular network. You can stain and label for lots of other proteins that you would expect to see in the bone marrow. This is stem cell factor labeled here in green. It's not necessarily the same in both of the niches. It looks like it's more dominant in the perivascular side. Here's a blow-up view of that. You can see really nicely the vascular networks now in these images with this confocal image. Here you see the vessel in cross-section where my laser pointer is here, as well as over here. You see the vessels in cross-section. So much of this, all this black area right here is actually the lumen of the blood vessels. Stromal-derived factor, or SDF1, also CXCL12 is a very common, well-known factor that's present in the bone marrow. We also see that more in the perivascular niche here, shown in green. And this is one of my favorite pictures that Drew got. It's just kind of visually very attractive, and it's basically a stain for laminin, showing the endothelial cells as well, laying down the basement membrane very nicely. You can see it over here. So again, all of the black here are the vessel lumens in this confocal image. So do we have hematopoietic stem cells, and are they differentiating into leukocytes that you would expect? This is just the fundamental function of bone marrow. So this is a confocal stack. It's really messy looking. And I think if you get into bone marrow or you want to study it, you realize the cellular composition is super messy. This is sort of a normal histological image down here. You see bone in pink here, but this is all of the cells. They're just all jammed together. It's very difficult to study. It's different than other organs where the cellular structure may be much more organized. We have a number of different labels here. CD34 labels the hematopoietic stem cell, I mentioned that, but it also will label the endothelial cells as well. So you can see the vascular network. CD33 is a more stem-like marker. The dark blue is actually sort of a pan-leukocyte marker. If you can see these, it's a pan lineage. It actually marks for a bunch of different leukocytes that have been differentiated. And then light blue is DAPI, so you'll look at all of the DAPI nuclei. So the different niches are messy looking. I highlight this here in yellow, and I'll show you what the confocal stack, the movie looks like. And this is really still my favorite image. It really highlights where my laser pointer is here. This is the niche that we've been trying to create. It's beneath the endothelial cells. And you'll see what I mean by it's a niche. It has all of the cells sort of present in bone marrow. So this is a movie that runs through that. And the markers are over here to remember CD34 is a hematopoietic stem cell. And you'll see the black where my laser is. This is actually a vessel lumen. So this is the niche itself right in here. And you see all the different cells that are in there, including the dark blue represents the different leukocytes that are differentiating from that hematopoietic stem cell. And you see the presence of the hematopoietic stem cell as well. I'll let that run through one more time. You see the CD34 positive hematopoietic stem cells in the niche. And then I think I have a still image that's maybe right about, maybe there. Yeah. So this is the still image here. And again, the arrows are just showing, here's a leukocyte that is differentiated. Here's the hematopoietic stem cell. And here's what normal bone marrow smear would look like as well. So when we take those cells out, I mentioned we can digest and do flow cytometry on them. So these are just some classic markers, myeloid, erythroid, and lymphoid lineages that we see. And I won't go through the flow cytometry in great detail, but we see all three of those lineages present out to day 14. That's really as long as we've taken this out, is about 2 weeks. The Methocold assay, remember the poor man's version or poor person's version of the bone marrow transplant. You take these cells out and you culture them and you look for different shaped morphology of colonies. And we're really interested in what's called the gem colony, the granulocyte, erythrocyte, megakaryocyte, monocytic. So it's a colony that has multi-potent potential or indicative that you have the hematopoietic stem cell. And so if we look from day 7, day 10, day 14, we see the presence of these gem colonies consistent with a viable hematopoietic stem cell out for 2 weeks. And this in the bone marrow field is a very big deal. If you culture these cells, these HSCs, hematopoietic stem progenitor cells, just in monolayer culture, they rapidly differentiate and you can't maintain them. So I think this is a pretty centrally important feature of our model. I like to show this video. It shows, it's a time lapse that I think runs for maybe 12 hours. It was sort of an overnight. It just shows you kind of how dynamic these cells are inside this bone marrow model. They're just jumping and migrating all over the place. Mostly why I show this is you can actually see the cells trafficking right here where my laser pointer is in the bottom left, right? So if this is our bone marrow, the cells can actually enter and exit through the fluidic line right here. So this fluidic line then becomes a model or a, I guess a model is the best term, of the peripheral circulation. So we don't have to digest the cells and take them from the marrow. We can actually look at the cells that are actually leaving the marrow as a possible indication of what you might see in the peripheral circulation of the peripheral blood. So these cells tend to be predominantly neutrophils. So they're CD15 and CD33 positive. They also express CD66B, which for those of you that are interested, that's a more mature neutrophil. So we're seeing predominantly neutrophils. And again, if you study blood, neutrophils are actually one of the predominant cells your marrow makes because neutrophils in your peripheral circulation have such a short lifetime, maybe 2 to 3 days. Whereas red blood cells, although they're the much more dominant cell in your peripheral circulation, they live for 30 to 60 days. So your bone marrow is actually making many more neutrophils. And that's really the problem if you are thinking about compromising the bone marrow with chemotherapy or radiation or something along those lines. Neutropenia or the loss of neutrophils is really the biggest issue. So I mentioned this lower chamber here. Once the bone marrow model is made, we can load cancer cells in here. So we're just beginning to explore this. Here we've loaded MD MB231. This is a classic breast cancer, triple negative breast cancer line that's constitutively expressing GFP and MCF7, which is a estrogen receptor positive cell. And those are thought to generally go to the bone marrow and become dormant. So these are just demonstrating proof of principle that we can load this bottom chamber, and you can see the green migration of these cells up into the bone marrow compartment. We haven't really done anything specifically seeing whether or not they become dormant or not yet, but that is something that I remain very interested in. I'll just show you quickly one thing we've done with it. We got a small pilot grant last year to irradiate the bone marrow. This was actually from a sort of a branch of NASA. They're interested in radiation exposure to astronauts. So we have a proton beam at UC Davis that can deliver radiation doses. And again, I don't know a lot about this, but the radiation physicists and radiation biologists have really helped me out. So we basically exposed this bone marrow to a dose response. And it's a slightly different model. Here we are doing just the perivascular niche. You can irradiate this, take the cells out. Those cells that are exiting, I think we call them circulatory cells that I show here. Those are the ones that are actually leaving, but we can actually digest and look at the cells on the inside as well and do flow cytometry. This one was such an interesting project. This is probably the last thing I'll be able to show you today because it took such a large team effort. In your own lab, you know, everyone has their own individual project, and sometimes very large projects can be difficult to sort of manage. This one involves 6 different people in the lab because of the complexity of making so many devices. So we actually took a picture. I think we had 165 of these floating devices for this one radiation exposure. There's a couple of busy grad students, and basically we turn the device on its side. We only have to keep it on its side for maybe 20 minutes, and we can irradiate it with a proton beam. And I'll just show you kind of just really simple results. We're showing here DAPIs in light blue. The dark blue is the leukocyte marker, and you see white blood cells as CD45 marker in green. This is the control. And then when you expose it to 4 gray, you see this sort of obvious drop off at 4 gray. It's a pretty noisy assay. You see the noisy, but here's the dose increasing and the CD45 numbers slowly falling. As well as the neutrophils or the CD15 marker almost disappearing once you get to 4 gray. Perhaps most interesting were the cells we actually collected that exit the bone marrow. And here we see potentially a big drop off right at 1 gray for the neutrophils. So this becomes really problematic, maybe a sign that you're modeling neutropenia. And 1 gray, although it's a very convenient number, it's 1. That actually is thought to be the radiation dose that an astronaut might be exposed to on a trip to Mars and back. So 1 gray was definitely in our dose response. So MK, I think I'm going to end there. I'm not going to be able to tell you about primary human organoids. We've loaded primary breast and colorectal cancer into these devices. We've looked at angiogenesis. We can put in white blood cells and watch them extravasate. Here you see them extravasating from the blood vessel network into the tumor microenvironment. I just always run out of time. And these are some basic science questions that we looked at, where we looked at migrating tumor organoids. Priscilla was in the lab at the same time MK was, and her paper came out in Cancer Research. So you can see these are mouse breast cancer organoids. So you can use these fluidic devices to watch them migrate in response to different gradients, in this case, SDF1, and ask, this is not to model patient-specific cancer, but this is to get at some basic science mechanisms. So I'm gonna wrap it up there. And you can read my concluding slides, but I always want to leave some time for questions. So let me just end there, MK, and hopefully people are typing in some additional questions in the chat box, and I'm happy to answer some questions.
All right, thank you so much, Steve. That was really nice to get to see updates from everybody's work. So anyone who has questions about any parts of the talk, feel free to throw them in the chat box at me, and I will do my best to read them and communicate them. We have a question about the basic microvascular systems, and it was, scroll back up. How full, I guess, or large maybe do the vessels get in the diamond devices so that they survive the full 28 days? Or do you see changes, I guess, in kind of the size and fullness of your cells as you take it out farther?
Yeah, it's a good question. I took out a slide that I show sometimes. It kind of shows what people would consider vessel network remodeling, or pruning is sometimes the term that's used. So the vessels never tend to get thicker than about 100 microns in diameter. I think that one picture where I paused earlier, there was a kind of a big vessel running down the center that was maybe 75 microns. So what usually happens is most of the vessels will tend to regress, and you'll have a small number, maybe 3 to 5, that tend to sort of dominate most of the flow. And I think that's just a reaction to the microenvironment that you have in there.
Awesome, thank you. We have a question from an undergraduate student who's looking to apply for PhD programs. And he wants to know kind of where have your students and your trainees ended up over the years?
Yeah, sure. Well, I've been around long enough that they've pretty much headed everywhere. Actually, it's kind of an interesting question. I think there's probably 8 or 9 of my trainees that have gone into academic positions. MK is certainly one of the more recent ones. Many have gone into the private sector. Some have gone on to med school. So there's just been so many But because, we're in this sort of virtual environment right now, I actually invited 3 of my alumni to come back and visit during my group meeting, which is weekly. And yesterday, we were visited by Monica Moya, who actually
Nice.
was the key trainee to sort of develop these vascular networks. So I'm going to continue to ask trainees back to talk to the lab about, their career paths and what they've done. And if we stay virtual long enough, MK is going to get an invitation from me as well.
I'm still owed an actual trip out to your house because that was
Yeah.
supposed to happen and it got canceled. So
Yeah, I figured I wouldn't pester her too much since she's trying to launch her own lab this year, but eventually I'll still get an invitation.
yeah, thank you. I had a question about the bone marrow environment. In Drew's laminin picture, it looked like there was more laminin in a part of the chamber than another part. Is that maybe due to the flow properties, or is that something that was really noticed in any other devices, or just an artifact?
Yeah, I would imagine it's sort of representative of the just general heterogeneity that you're going to find in the vascular network. That's sort of the easy answer. But these vessels are not going to be really exactly the same. It could also be potentially an artifact of the staining. You have to wash these antibodies in and wash them out, as MK knows. And sometimes you get beautiful images. Sometimes you get some artifact from them as well. I don't know. When we actually publish that, it's in the paper that's almost about to be accepted. One of the reviewers wanted us to quantify these changes, but we kind of pushed back, and we just didn't think that the model and that particular endpoint, it really made sense or was going to offer anything to try to quantify it. I think we're just going to see some variation in the level or the amount of laminin that you're going to see in these vessels.
Sure, makes sense. Thank you. Any other questions about the bone marrow, about microvasculature, collective migration, tumor organoids? Steve kind of does it all, so. I'm not seeing anyone else, so let's all thank our speaker. If you want to, we'll use a little clap emoji, you can, or turn your video on, however.
Yeah, it's always slightly awkward. Let's thank the speaker, then you can't actually hear any clapping. It's fine. I see some thumbs up.
Oh, good.
It's been really fun, you guys. I hope it's been enjoyable. It's the best we can do. I really would have preferred to be there in person, but we're just not. We're living in a different world right now. But thank you very much for your attention.
We're glad you could fit it in this way, because I know how busy you were in the before times and how much you travel. So I'm really glad we got to include you in this series.
Well, thanks you guys very much, and enjoy the rest of your day. And hopefully I've inspired you somewhat to maybe work in the field of organ-on-chips.
Okay, thank you so much.
Thanks.
Bye.
Bye.
Bye.